Hello, everyone. Today I'm going to talk about a method that we worked on for analyzing cellular communication, given some spatial annotation of transcriptomics data. And alongside that, I'll also talk about some related methods we developed that mainly serve as some auxiliary method to assist the analysis of this cellular communication in spatial transcriptomics. Okay, so I guess I can skip this introduction to single cell biology. It's basically uh, recently after this single cell RNA-seq, now we have people have extended along the direction of omics. There are multi-omics now, considering, for example, proteomics and ataxic. And people are also extending along the spatial direction, where we could have some spatial annotation of spots or cells in the intact tissue. So this spatial transcriptomics is really motivating us to develop some more, I would say, more detailed analysis of cellular communication, because cellular communication is, especially the ones through contact, cell memory contact, or diffusible ligands, are really restricted in localized in space. Okay, and of course there are two pre-processing steps, I would say, that we want to handle before we really apply some cellular communication analysis. First one is, as we know, for example, you're familiar with cell chat, right? So this, when we consider cellular communication, is really more, is usually more reliable to look at populations or groups of cells rather than individual cells. So in spatial transcriptomics, it's probably better, intuitively, it's probably better to group the cells based on both their cell type and also their localization. It's considering a spatial group like this and another spatial group like this, there could be a local cluster of cells that express the receptor and a local cluster of cells that expresses the ligand. If we take this traditional grouping of cells, it's likely that the average expression of this receptor in this group and the ligand in this group are both weak, but they actually assemble some strong communication, local communication connections. So this is something we're working on, trying to get a spatial segmentation or grouping of cells that are more suitable for cellular communication analysis for spatial data. And another thing is we're also trying to see if we can integrate multi-omics to also aid in better analysis of cellular communication. Where spatial transcriptomics alone, many technologies is like the gene expression resolution is usually inferior to the single cell technologies. Okay, so I will start now into some methods. I will try to give intuitive descriptions of what all these mathematical formulations are doing. And please stop me whenever there's a confusion. Okay, so for spatial segmentation of spatial data for cellular communication analysis, basically we're thinking of deriving some low-dimensional representations of spatial spots in spatial transcriptomics data, such that these representations will encode both the gene expression profile at one spot and also its spatial neighborhood. Okay. So this, as an example, we can, given spatial data, which is basically a point cloud in space, we can build a spatial graph. For this particular work, we're building a alpha complex based spatial graph, which we found to be more effective than the traditional KN graph. that better reflects the local geometry of the tissue. And then based on this spatial graph of spots, I think like many methods in this direction, we can build a graph neural network such that according to some loss function, we derive a low-dimensional feature that captures the key information on the nodes of the graph, as well as its neighborhoods on the graph. Okay, so basically what we're trying to do with the loss function is we want a feature vector that captures both information, unlike PCA in single-cell data that only captures the information on each individual cell. Then once we get this low-dimensional embedding, we're able to feed it to some clustering algorithm to get a spatial segmentation of ST data. So as a result, with this simple method, we can get a pretty good segmentation of spatial data of structures at different scales, different spatial scales. Okay, and then we wonder, for cell-cell communication analysis, of course, cell-cell communication analysis is one of the goals for this line of work. We wonder that if we want to get some spatial localized clusters, for example, the example I showed, for this type of structure, for cell-cell communication analysis, we probably want to separate it into two spatially localized groups so that it's meaningful to take the average expression and look at the communication strength. For this, we simply thought that based on the previous work, we introduce one additional simple loss term such that despite these formulations, intuitively, in addition to a feature vector that describes the gene expression at spots and its neighbors, we promote a low-dimensional embedding such that two spots will have similar feature vectors, only if they're both similar in gene expression and also close in space. So this low-dimensional embedding, we can first test it in traditional tasks of tissue segmentation. This method is what we call the space flow. We see it has a pretty good segmentation of the spatial tissue, and it also, because of the additional loss term that we promote a smooth embedding over space, we're able to show some smooth transitions, spatially smooth transition of properties in the spatial transcriptomics data. Right now, we're also trying to develop some tailored clustering algorithm based on these low-dimensional embeddings we get, to derive some more suitable clustering algorithm to be used with cell-cell communication analysis. Okay, so these are some preliminary work that I hope that can help us prepare better input data for cell-cell communication analysis. Okay, so for the main topic today I'm going to talk about is one dominating mechanism of cell-cell communication, basically the cell-cell communication through diffusible ligand, where some cells will produce some proteins that are water-soluble that can diffuse in the tissue. And then there are also some cells that express membrane proteins. Many of them we call the receptors that can bind to some of the compatible diffusible ligand. And once a receptor receives a compatible ligand, it will cause some structural changes and probably trigger some downstream processes inside the cell. Okay, and before that, one additional pre-processing we can keep in mind is for spatial data, we can often increase the quality of gene expression data by integrating some single cell data. For example, in this extreme case, even if we don't have really spatial transcriptomics, but instead this very traditional imaging data of just a handful of genes. Then we can try to integrate single cell data into this imaging data or some low quality spatial data to try to predict or recover the gene expression profile at the transcriptomics level. Okay, we'll keep this task in mind, and I'll show you in a moment that the methods I'm going to talk about, particularly optimal transports, will be used for both tasks of cell-cell communication analysis as well as this integration of datasets for better quality. Okay, so this tool I'm going to talk about is called optimal transport. And before we go into the details of the tool, let's go through some simple examples that we hope these methods could capture. So, for example, in this first simple scenario, let's imagine that we have 3 cells placed on this one-dimensional tissue, this artificial structure. And let's assume that the blue cells are the cells that, let's assume we only consider one pair of ligand receptor. And the blue cells are the cells that only produces ligand. And the red cells are the cells that only produces the receptors. And we have gray cells that are this background cell with no ligand or receptor produced. So in the first scenario, if we have one ligand expression cell here and 2 receptor expressing cells here, because this red cell on the right is farther away from the cell on the left, from this blue cell. So if all the amounts of receptors they produce are all the same, then we desire a method that assigns a stronger edge weight to this connection than this connection. Right? And versus the second case, if in the middle, the cell in the middle does not express any receptor, then we desire a connection here whose weight is between this higher weight edge and this lower weight edge in the first case. Okay. So you see here, using some traditional graph-based scoring system. For example, we can always design an empirical function that takes into account the expression level and also the spatial distance that outputs a score. It won't be able to distinguish between these 2 scenarios, such that when the cell in the middle becomes a cell that does not express the receptor, the edge between the 2 cells on the 2 sides should have a stronger weight than this edge in the first case, due to no. So in other words, we want to somehow capture the competition between cells for the same compatible molecular species. Okay, so this, in summary, is competition between cells. And let's consider this a second scenario where we also have the same 3 cells on the same one-dimensional fake architecture. And let's say in this case, we consider 2 ligand receptor pairs that share the same receptor, which is a quite ubiquitous scenario in practice that these ligands and receptors are also exhibit some multi-compatibility among species. So in this case, assume that both blue and green cells produce the 2 types of ligand that can both interact with the receptor produced by the red cell. So in this particular case, if all the binding affinity are the same, if all the molecular amount are the same, then these 2 edges on the top should have a very similar weight. And when the green cells, in the bottom case, when the green cells are absent, right, that means in the second case, there's no competition in this simplistic example, right, for the blue cells to communicate with the red cell. So in this case, we want this edge to be stronger than this edge, than the same edge in the first scenario, right? So in summary, this is another situation that we were considering that is basically the competition between molecular species due to the multi-compatibility among this ligand and receptor species. Okay, I want to pause a little bit here to see if there is any questions about the general goal we want to achieve with the computational methods. Okay, so let me give a very brief introduction of, or a brief intuition of what optimal transport is doing. So I think it's helpful to think about optimal transport as a generalization of pairwise approaches. By pairwise approach, I mean you're given two cells, then the strength, the connection between two cells, for example, in the cell-cell communication case, does not depend on the interaction among these two cells with other cells. It's purely defined on these two cells alone. So in other words, when you derive a score for a pair of cells, these pairwise approaches like assigns scores independently for each individual pair. And optimal transport, we can think about it as a generalization of this idea to a global perspective, such that when we consider the interaction between two cells, we at the same time, we also consider their interactions with other cells, like a third cell or a fourth cell. In the cell-cell communication case, you can see here there's only a limited ability for the red cell to take ligands because of the limited amount of receptors. So the strength of communication between the red cell and the blue cell also depends on whether there's some other cell in the neighborhood that produce the same compatible ligand. Okay, so as a illustration of what optimal transport does, is we're given two distributions. We can think about this x as a pile of sand, and this y as a destination of the sand that we want to basically we want to transport this x, this pile of sand, to fill the hole in this region represented by the distribution y. Okay, and there are different ways to transport this mass, right? We can transport, for example, we can transport this bunch of sand to this location, right? And this bunch of sand to this location. Okay, and each transportation is attached with a cost. In this particular case, we can assign the cost to be just the distance in this one-dimensional axis. And that's noted by the cost function c of xy between every pair of locations. Oh, sorry. Okay. So now optimal transport is trying to derive a transport plan such that the total cost is minimized. Okay, so basically there are, for that pile of sand example again, there are infinite way of transporting local sand piles to the local holes. And we want to find the optimal way. By optimal, we mean the total cost is minimized. So basically, let me go back to this example. So if you attach in the discrete setting, okay, if we attach a grid system to this two distributions, then we can use a matrix to annotate how much sand we transport from one location to another, right? So we can think about this as locations in X and this as locations in Y. Between each two locations, we can assign a cost value. For example, cost equal to 2, cost equal to 3 here, okay, based on their spatial distance. And we use M matrix to denote the cost matrix. And what we want to find is the transport plan. Okay, it's the same dimensional matrix. as the transport cost, except the meaning of the elements in the matrix are different now. So if we have, for example, point 1 here in this location, that means we have a transport plan that transports this amount of sand from this location to this location. And then the total, the cost of transporting this amount of sand from this location to this location is based on both the amount transported and also the ground cost. Okay, so in this case, the cost in this particular location would be 0.3. And you can imagine that we add up all these locations in this two matrices multiplied. That gives us the total cost of transporting the entire pile of sand to this entire whole structure here. Okay. And also we want that the amount we transported does not exceed or matches the marginal distributions. Right. So at this location, you only have this amount, for example, the amount of 1 of sand. Then all we can transport from this location should be the same as this number here. So in other words, we want the summation of this row to match this number. Okay. So all these descriptions can be summarized into this basic formulation on the left, okay, where we want to minimize this cost function. That's the two element-wise multiplication of matrices summed over all elements. And we want to have the constraints such that the marginal distributions matches the original given distributions. And we call this matrix gamma the optimal transport plan. Okay, so that's the original setup in traditional optimal transport. And basically, we require 3 pre-given stuff. One is the two distributions, alpha and beta, and one is the cost matrix M. So we can understand this as alpha and beta are two distributions embedded in the same space that you can compare every pair of locations. And in practice, it's less desirable to have this a very strict marginal distribution constraint, right? So like these two constraints here, that exactly matches the sand you transported exactly matches the original pile of sand. In practice, people can relax this marginal mass conservation by changing this strict requirement into some basically some soft penalty. Okay. And in some other cases, we don't even necessarily have two distributions embedded in the same space, that we cannot directly compare two locations from the two distributions. So instead, what we're given is just a intra-dataset distance. So for our biological examples, we can think about one single cell RNA-seq and one single cell ATAC-seq data with no correspondence between cells known. And also, let's assume we don't have any information between the correspondence of the open regions, the open locations, and also genes. In that case, what we have instead of a inter-dataset distance is we have two intra-dataset distance matrices. So basically, in this case, we can also use optimal transport. Intuitively, what it does is it tries to match the two structures of the data set. Okay. Imagine that we have a data set like this and a data set like this embedded in two different spaces, right? For this simplistic example, we don't need to know the correspondence between each pair of locations, right? Instead, we can just try to match the two shapes, the two global shapes, to integrate them. So that's the idea of this so-called Gromov system optimal transport that is applicable in more general cases when we don't have an inter-dataset distance matrix. Okay, so as a discrete example, these edges are what we get when we compute optimal transport plan. Consider this alpha are the pile of sand, or in some other intuition, we can think about it as locations that produce some product. And the beta, the blue dots, are locations that demand the product. And then there's a transport cost between these locations due to this Euclidean distance in the two-dimensional space. Right. So in other words, we want to find connectivity plans between these red points and blue points such that all are connected. And also the total length of these edges are minimized. Okay. So that's the traditional setup of optimal transport. And there are scenarios that traditional optimal transport doesn't take care of. I'll give two simple examples here. First is, imagine that our ground truth may be like this. This is a plot I took from Scanorama. So imagine that the ground truth is this, but in practice, what we have, instead of viewing the entire ground truth, we have two partial observations instead. And in this case, we don't want a complete integration of the two data sets. In other words, we want to only match part of the two data sets. And in some other scenario, for example, in cell-cell communication, let's assume this blue and red distribution represents ligand and receptors respectively. Then this simply for some pair of locations, for example, this pair of blue and red locations, if their distance is too far away, we simply want to exclude the coupling between these two locations, right? Due to the unreasonable signaling distance. Right, so summarize these two scenarios. Basically, we want to avoid some couplings in the transport matrix. Or in other words, we want to exclude certain edges in the final transport plan, which are unreasonable in our biological applications. Okay, so that's, I probably want to skip the details, but that's basically changes the traditional optimal transport. It becomes a very different problem due to this potentially complex additional constraints on the elements on the transport matrix. By the element-wise constraints, I mean the locations, the connections on the transport matrix, which we think is unreasonable for in the biological setting. So this becomes, unlike traditional optimal transport, this becomes a double layer optimization problem, which we derive into a we prove that we can achieve a simpler equivalent form, from which we can derive efficient algorithms. So just for an idea of the efficiency of these algorithms, we're able to look at like 1,000 pair of ligand receptors at the same time, considering all the competitions among them, using a medium grade laptop to compute in a reasonable amount of time. Let me skip this additional. So basically, these are also showing that the new methods also has pretty good mathematical utilities in general applications. I'll skip this. So as a result, what we see is, again, we have the two distributions we want to find connection of as light blue and light red. And this solid blue and solid red are the coupling given by the optimal transport problem. Okay, so compared to traditional optimal transport, we have two utilities. First, we can control the cutoff so that it's guaranteed that the result, every element in the transport matrix will have a cost, element-wise cost, that's bounded above by the given cutoff. And for example, in cell-cell communication analysis, this cutoff could be, for example, 200 micrometers. That's if you think any communication through diffusible ligands exceeding this distance is unreasonable. And another good property due to the elimination of the marginal distribution, strict marginal distribution constraint, we allow this partial coupling between distributions. And as a result, in traditional optimal transport, we need the two original distributions to be probability distributions. In other words, this light blue and light red have to have the same total mass. Now, in this framework, we don't need that anymore. So the advantage of this is we can keep the original unit, right? So imagine that there could be a ligand that's expressed only in a localized spot, and there could be a receptor expressed everywhere. So if we normalize them to be probability distributions, it disturbs the original unit a lot, which is what we don't want. So as a intuitive example of what we're trying to do, this is a classical example. In this task, we're trying to produce some image that basically have the pattern given in the source image, but with the color palette from the target image. Basically, using this supervised optimal transport, we're able to control how much color we transport from the target image to the source image, or how much color we keep for the source image after the transport. So this is pretty important for cell-cell communication analysis, that if two distributions of ligand and receptors are too spatially far away, we simply don't want to transport anything. Right? So, but like these traditional approaches, the colors are always all transported to the source image. Okay, so that's the background of the method. Now back to our application, we're able to use this this optimal transport method to integrate, for example, single cell data with spatial data, both transcriptomics, using the setup with a cost between two data sets. And for more challenging scenarios when we don't have shared features, of course, there are ways to turn this ataxic into a gene activity matrix and somehow relate to the gene expression columns in the single cell RNA-seq data. But in this example, let's assume we don't know correspondence between the features, and we don't know correspondence between the cells as well. But in this case, we can also use a Gromov-Wasserstein optimal transport to connect two data sets to achieve some basically multi-omics data sets that can potentially help with the cell-cell communication analysis. So intuitively, as a result, what we're doing in this scenario is we can match the global structures of two data sets. Okay, while preserving the local structures that may be captured in only one data set. We'll show this in this simplistic example. So if you consider this black and red data set, globally, they're both like a long string structure. But for the red data sets, there are some local features. For example, there's two small loops on the end. And as a result, we're able to integrate the global structures of the two data sets and also show this tiny detail structures that was only present in one of the two data sets. Okay, so these are some method background and some pre-processing we can do to theoretically enhance cell-cell communication analysis. Now back to the main problem alone. The two key features that we consider is one is that there are competitions for molecular species, right? Due to this multi-compatibility between ligand and receptors. For example, this is an example with 3 ligands and 4 receptor species. one ligand can interact with several receptor species, and the same one receptor could take different species of ligands. For this, we can show that we can turn this into a huge optimal transport problem, considering all the molecular species at the same time, by introducing these large blocks of infinity values in the cost matrix. So as a result, we can see here, this is the effect we get. That basically when we consider each pair of ligand receptor interaction, we're also capturing the competition between the ligand receptor species. And we also validate this using some simplistic PD model of cell-cell communication that captures the basics of this process, like the diffusion of ligands, binding dissociation of ligand receptors, and also the degradation.
Hi, Zixuan. Sorry to interrupt. We have one question in the Q&A.
Yeah, I saw the question. Yeah, that's a good question. So I think this depends on what we're trying to study. Sometimes if the focus is more on where the communication happens in space, then it's probably okay to directly apply to Visium data. But in most cases, you also want to explain what types of cells are participating in the cell-cell communication. In that case, a straightforward way is use whatever decomposition methods that are out there, and then reapply. And then, like if you find 3 types in one single spot, then we can make pseudo spots at the same location, but 3 spots each represent one cell type, or even one single cell. And then this optimal transport will apply in the same way to the data. So I think back to a question, if we care about the cell type, care more about the cell type than the general location of communication, then we can always use I would say most of the decomposition methods or deconvolution methods that are out there, and then the results we can directly feed to this optimal transport framework. That's the resulting optimal transport would have interpretation of cells or cell types. Hope this answers the question. Yeah, let me resume. Okay, so in this benchmark or synthetic benchmark, we can see that the OT approach agrees very well with the very detailed partial differential equation approaches. We also validate this in a lot of randomly generated patterns. Basically, in real analysis, we can imagine that there are thousands of, for example, in cell chat database, there are over 2,000 ligand receptor pairs, right, and hundreds of species. So it's very computationally costly to model this via detailed partial differential equations, but our optimal transports framework could compute for all these species at the same time relatively efficiently. As a result, I think related to the question we had, sometimes people are interested in the directionality of cell-cell communication, right? How it corresponds to the global patterns of tissues. In that case, we can probably directly apply this method to the raw spatial data without any detailed annotation of individual cells to get this what we call the spatial directionality of cell-cell communication. So how we get this is similar to SC single cell velocity. It's like when you get this communication matrix, it's a cell by cell matrix, right? Then instead of interpolating this matrix as a vector field in the string expression space. Now we interpolate this matrix, this connectivity matrix, in the spatial space. Then we can have an annotation of this directionality of signaling. For example, in the bottom, this wingless signaling was known to shape the horizontal pattern of Drosophila embryos, and the DPP signaling was known to shape the vertical patterns of the Drosophila embryos. And we're able to capture these two global directionality of cell-cell communications. And also, again, if we only care about the location of where cell-cell communication happens and the general directionality of the cell-cell communication, we can directly apply it to Visium data. And of course, if you apply it to higher resolution data or Visium data with decomposed cell type or even mapped single cells, then we can have additional interpretation of the communication results. But basically, these are the main outputs of the method. For each individual, let's say we have a list of ligand receptors that could have multi-compatibility. For each ligand receptor, we have one matrix. Let's use this. And this matrix is a n cell by n cell matrix, such that the ijth element of this matrix simply means the signaling strength from cell I to cell J through the ligand receptor pair, in this particular case, through the pair L1 and R1. And we get this matrix for every ligand receptor pair. Okay, so if we sum over the rows of this matrix, we get the total amount of received signal by each cell. And if we sum up over the columns of the matrix, it gives us a sense of the total amount of sent signal from each cell or from each spot. Okay, then from these summaries, we can then use some existing method to try to find correlations between the cell-cell communication process and some potentially downstream genes. For example, this is the result got from the method TradeSeq. I think it's a popular method for finding the temporally differentially expressed genes given some annotated pseudotime of cells in single cell data. Right, and now assume that instead of using pseudotime as the cofactor, we replace pseudotime by the amount of received signal by each spot. We can run the same analysis and find the genes that are highly correlated to the cell-cell communication processes, okay, through potentially nonlinear correlations or nonlinear relationships. So these are more like a correlation analysis, and we could also try to dissect it more using this partial information decomposition. And basically, we can use and this partial information decomposition is a long-existing method. It basically captures the information that can be told. For example, in this case, the information that can be told by Y about Z, given the information of X. Okay, so in our application, we care about the information that can be told by one particular cell-cell communication process about one particular gene of interest, given the background genes in the same cell. Okay, so that's one more step of finding the more unique, I would say the unique correlation between cell-cell communication and gene expressions. And in a more, I think a more general case, we could also use, so this partial information decomposition, it quickly becomes computationally costly when you consider more than two background genes at the same time. Okay. And by background gene, I mean when we consider the impact of L1R1 on gene X, given the information about gene Y. If there are two genes in gene Y, then it becomes computationally costly. So for higher order interactions, we could use a more strict, more black box tool where we try to predict the expression of a gene given two types of features. One is the cell-cell communication we inferred, and the other one is other highly variable genes or other genes that are correlated with the genes of interest. Then we look at how much prediction benefit we get by adding this cell-cell communication as additional features. Right. So intuitively, if the improvement we get is a lot, that means the cell-cell communication we inferred is providing a lot of unique information about the genes of interest. Okay, and in this case study, we used this approach and identified a bunch of genes that are likely related to the TGF-beta signaling that we, in the first place, inferred to be highly correlated to the spatial temporal development of human skins in this particular data set. So basically, in this particular data set, we run this cell-cell communication analysis. We get a lot of directionality of cell-cell communication. We find that the TGF-beta signal interaction agrees very well with the the known developmental trajectory annotated by the pseudotime projected to space. Okay, then we think this TGF-beta signaling is maybe highly related to the development of skin. And based on that, we can further use this machine learning approach to screen over genes that are likely directly related or either upstream or downstream with respect to this TGF-beta signaling. Okay, so we call this cell-cell communication DE genes. So that's mostly it for the method. And I think I'll give an example of the code interface. So basically, this method requires two things. One is a given ligand receptor database. In this particular example, we're using our familiar CellChat. And it's basically we get basically a AN data with annotated spatial distances. Okay. In the default setting, we just use a straight line distance. But sometimes we can even define a custom distance matrix, for example, considering the tissue architectures. Some tissue regions are more dense and but denser than other tissue regions, making the diffusion with a shorter spatial range. For that, we can also customize the spatial distance to reflect that. And then we can run the communication. And like I said, it produces one main output for one particular ligand receptor pair, just a cell by cell or spot by spot matrix. And from that matrix, we can turn it into some other interpretable objects, like the total amount of received signal, the total amount of sent signal of each spot. And we could also interpolate a spatial vector field describing the directionality of cell-cell communications. And of course, we could also, that worked, the communication network was computed at single cell level, but for more reliable interpretation and display, we could also always summarize it to cell clusters.
Sorry to interrupt, Dr. Cang. We might only have 2 or 3 minutes since we will save some time for the Q&A.
This is the last slide.
Okay, great.
Yeah, and we can also do some downstream analysis as we did, find this cell-cell communication DE genes. So this is like a pseudotime DE genes, except the horizontal axis becomes the strength of signaling. And we can also use some machine learning method or information approaches to look at this higher order interactions between cell-cell communication and gene expressions. So as a summary, we talked about a method, COMMOT, for inferring cell-cell communication from spatial data. And we also talked about several methods. This scannet space flow for potentially better segmenting the spatial data for cell-cell communication analysis, and also two methods for integrating this omics dataset for potentially better quality datasets for CCC analysis. And motivated by all these biological applications, we also, along the way, we developed this two optimal transport, which also turned out to be mathematically interesting. Okay, and with that, I would like to thank my collaborators, Dr. Qinye, mostly on the biological aspect, Dr. Yanxiang Zhao on this optimal transport on the mathematical objects, and Honglei Ren and Catherine for the two, for spaceflow and Vida, the two computational methods we developed. Thank you for your attention.
Great. Thank you for your talk. So if you have any question, please type that question in the Q&A. Maybe give other audience some time to think about the question. So I actually have one or two questions here. If I remember that correctly from your paper, you actually test your software COMMOT in different spatial transcriptomics data set like Vism, Murphy, several other cell types also. So could you let us know? So for the COMMOT, which data set actually have the highest accurate score? Like basically, if we are going, say for example, if we are going to do the spatial transcriptomics analysis, well, we are going to prepare this kind of library, and we want to use your tools. Which data set do you actually recommend?
In my experience, I actually find this generally Vism data works well due to its better coverage of genes. And of course, for Murphy, you might not be able to look at all possible ligand receptor pairs because it simply doesn't measure it. And for SlideSeq, this type of high resolution data with still near transcriptomics coverage, I find it usually the data is much sparser than Visium. And this optimal transport approach is on the detailed side of methods. So the sparsity would result in also sparse cell-cell communication output. And yeah, in my own experience, I find Visium data to be like more reliable to systematically look at all possible ligand receptors. And as the previous question suggests, we probably need some decomposition or integration with single cell data to get a better interpretability of cell-cell communications.
Great. So just follow-up question, when you say that we actually need to use some deconvolution method to do the annotation, like how the cell type annotation will affect the cell-cell communication analysis in the spatial transcriptomics data? Did you have this type of study in your project?
Yeah, I did it, but I have not published it. So basically, what you can do is you can consider one spot. Then after the cell type deconvolution, this spot can be turned into, let's say, 3 representative types. Let's use the size to annotate the composition weight. In that case, we can make I would say a pseudo spatial data such that these 3 cells or the 3 cell types are just assigned to the same location, but with different identities. Then after the communication analysis, if you add up the result for the 3 pseudo spots, they converge to the original results at original wisdom data. It's roughly, I would say, paired with the deconvolution method, we're able to also deconvolute the cell-cell communication matrix as well into the same level.
So does it mean like for each cell you have like weight for these kind of like 3 different pseudo cell types per se?
Yeah, ideally you want some weight to assign ligand receptors.
I have one question regarding like the computational time that you mentioned that it's kind of like computational heavy. So for example, if I have a data set, say VZIM data, 20K genes and 30K cells. So what will be the computational time that you can estimate?
For that, my experience is around 20 minutes. That's much longer than simple functions, simple pairwise functions. But I think it's
so you're saying 20 minutes, that's like a single CPU or like a multi-thread
like a laptop.
laptop? Okay.
Yeah, just a regular laptop.
Thanks.
Great. Any other question we have? Okay, seems there's no question. Well, thank you so much for the talk, Dr. Cang. Have a great day.
Thank you. You too.
Thank you.